In this presentation, I am going to talk about polymerase chain reaction or PCR. Polymerase chain reaction or PCR is an enzymatic process in which millions of copies of a particular DNA sequence can be produced. It is a significant process that can be applicable in both biomedical research and also in forensics. PCR, it was invented by Karry Mullis in the year 1983. This process is also known as molecular xeroxy. This procedure takes place inside the thermal cycler. PCR procedures have been simplified in recent years by the addition of only DNA to a pre-made PCR mix. There are several different PCR components needed for a PCR reaction. The most important part of a PCR reaction is a DNA template, a primer, DNA polymerase, DMSO, DNTP, and buffer. The other variables that can be included in a PCR reaction is given in the table. The most important part of PCR reaction is a DNA template. DNA template is the fragment that needs to be copied or amplified. Primer that consists of both forward and reverse primers. They are short oligonucleotide sequences that are chemically synthesized and are complementary to the DNA sequence. DNTP are the building blocks of nucleotides from which DNA polymerase synthesizes new strands. DNA polymerase aids in the addition of 4 different nucleotides to the reaction, that is adenine, thymine, guanine, and cytosine. Buffer provides the optimum activity of DNA polymerase by providing a suitable chemical environment. Magnesium chloride or magnesium ions provide specificity for the activity of DNA polymerase. Dimethyl sulfoxide or DMSO can be used in a PCR reaction if guanine cytosine content is very high in a template. So it aids in reducing the melting temperature of a DNA. Bovine serum albumin or BSA aids in preventing the addition of enzymes to the surface and also to the tubes. The next is thermal cycling parameters. There are different thermal cycling parameters and they can be chosen according to the hybridization properties and annealing of the template strand to the primer. Amplification process takes place inside a thermal cycler. After adding all the reagents along with the DNA, the PCR process takes place inside a thermal cycler with suitable temperature. There are different steps in a PCR reaction: denaturation, annealing, extension, and elongation. Denaturation is the first step. This process involves heating the reaction chamber for 20 to 30 seconds at a temperature of 94 to 98 degrees Celsius. Annealing takes place at a temperature between 50 to 65 degrees Celsius for 20 to 40 seconds, in which primer anneals to the DNA, introducing new DNA strands. Extension takes place at 72 degrees Celsius by adding DNTPs to the growing chain by DNA polymerase. And this is a stage in which optimum activity of DNA polymerase takes place. The final step is elongation. It takes place at temperature between 70 to 74 degrees Celsius to ensure single-stranded DNA template is fully elongated, and the time is between 5 to 15 minutes.